Welcome to Science Streams. If you are interested in all things science in a fun, wholesome environment, you are in the right place. SciAnts is a unique husband and wife team of Belinda and Lita, who hold their PhDs in molecular and systems biology from Dartmouth. What can you find here? The SciAnts team is dedicated to making science more accessible to our entire community. They believe that it is the responsibility of scientists to engage with the general public, and Twitch is one of their media doing this. By showcasing their own research, highlighting new discoveries, answering viewer questions, and sharing their passion with you. We do a variety of interactive science streams, including microscopy of ancient amber with insects, 3D printing, art, science news and deep dives, the occasional game, and more. If you've ever wanted to have a fun space to nerd out, now is your chance. We stream daily science content on Twitch. Understanding not just what we know, but how and why we know it is the essence of scientific knowledge.
So the nucleus, right, is here. The DNA is inside the nucleus, and you can see it's not a straight line. It's folded and jumbled all over the place. And so to figure out what they're like, what they're closest to, so they're not necessarily closest to the neighboring gene, right? The 3D space could be something like this, right? And then this gene is actually closest to this gene, right? Like the green and the purple are closest, but sequentially you'd think that the purple and the orange should be closest. But it's not necessarily the sequence that makes them in close proximity, but their 3D position. And that could also influence if the genes are on and off. Cross-linking is what they do. They stick them together with a molecular glue and then cut them, and then they'll sequence and figure out what was closest together. Like in mice, you can ask the same question. Like during development in a mouse, how are the cells different? Like, are the cells that are becoming your liver cells different from your pancreas cells and your brain cells? And is that connected to the overall structure of the DNA inside the cells at that time? And so yeah, with this, you get like the little circle. So to do the experiment, so if these, like let's say that purple region, is close to the green region, they stick together. So you glue that, you keep them glued together with formaldehyde, and then you fuse these ends together, so they make this nice little circle. They're still glued together, and then after you've done that, you remove all the waste material and things that didn't stick together from your solution, and then this is what you end up sequencing. And that lets you figure out the proximity of the genes and how they look in 3D space. You know, are they accessible? Are they open? Are they near other genes? Because there's also, this Chris is getting like extra complex. If I'm losing anyone, I apologize. This is getting to the idea of enhancers as well. So this green gene might not actually code for RNA. It might not code for like a protein. What it might actually do is, if it's in close proximity to the purple gene, it might turn on the purple gene a little more than it normally would be. It could also do the opposite. It could be a suppressor and be turning off something that's in close proximity to. So it's like the extra batch of complexity on there of, okay, like if you've identified a gene, Chris, that's like we said, different levels in each of these casts, you're like, it's this gene. There's probably other things that are acting on that gene that are making it change its levels. So it's like a signal cascade is what we call it, right? Like this turns on this, which turns off this, which if that's off, it can't turn on this thing, and it just keeps going and going and going. And so it's never like, if you find that one answer, it's not going to be that one answer. It's going to be 10 other things. And so like that's one thing that I was studying with the ants. The question was, okay, what makes adults behave differently? And so it turns out if you lower the soldier, that level of the gene in the soldier, down to what it is in the nurse, they'll start behaving like nurses. And so then the next question is, okay, if you turn off that gene and you do the same sequencing experiment, what other genes are acting differently? And that's how you then figure out how that cascade works.
Thanks for watching SciAnts Media, and be sure to hit those like and subscribe buttons to help our channel grow and support our science outreach and education goals. We can be found on Twitch most days of the week, and be sure to join us here for our next video. Thanks again, and stay curious, everyone.